Investigator Prize and is now on the board of the Society. To avoid any issues with background noise, all participants will be muted. Please use the chat box to submit your questions, and Keith will answer them at the end of the talk. As usual, this webinar will be recorded and available from the OTS website and YouTube channel. Right, over to you, Keith.
Okay. Well, looks like, hold on a second. Petra, do you have your mic on still?
Yes.
Okay, do you want to mute
Sorry.
yours just temporarily?
Yes, I was muting her.
Okay, so looks like you guys are joining me in my office, or maybe I'm hanging out with you wherever you are. So I was asked to give this webinar by Petra, and I've been working on chemical modification of CRISPR RNA. I don't do much of the modifications. This is collaboration with Masad Dama, who's been doing most of the synthesis. But we are interested in the structure-function activity relationships and outcomes on editing and enzyme activity. And so there's been a number of publications in this area, especially in the past recent 2 years. So I think it's a good time to review or overview CRISPR RNA chemical modification and what we've learned so far. That's kind of what this webinar is going to be about. So, most of us or all of us know a lot about CRISPR, and there's a lot of excitement about what it can do. For example, genetically modified organisms or GMO plants and products have really had a hard time making it into your produce section or the grocery store. And products because there's a big fear about the genetic modification. Something that CRISPR can do is it can really change that, because you can accelerate natural processes and select the right crops or the right producing plants without actually having to transgenically introduce new genes, for example. So CRISPR modified food would be considered non-GMO, and this is a huge thing for agriculture. Another area is in industry. So, for example, we use genetically modified bacteria to produce certain products, especially natural products that are hard to chemically synthesize. And so this is actually going to enable better industrial production of products and stuff. Okay, and of course, probably the thing that most of us care the most about is therapeutics, CRISPR-based therapeutics, and so they are sort of on the horizon, and we're hearing a lot about them. And this is an article that was just came out in the news end of February. CRISPR Therapeutics is starting its one of its first trials where it's infusing patients, and so this is going to sort of be the norm, I think, in coming years. We're going to hear a lot more about CRISPR Therapeutics. But there's some dark side to CRISPR, so to speak. It can have potential problems. One of the major issues is something we're all familiar with, is off-target effects. And so we certainly don't want to cause, trying to treat one thing and oops, we caused cancer or other diseases. So off-target issues is a big deal. Also, immune responses, delivery, efficiency, those are all concerns that need to be fixed for CRISPR therapeutics to really develop and grow. Okay, so presentation overview. Talk a little bit about why we would want to chemically modify CRISPR RNA. It may be obvious to some of us, but to other people not so much. Then I want to go through sort of almost one by one the CRISPR RNA chemical modification publications. There's only a dozen or so, maybe 13 or 14. There's not a lot. So it's relatively new, but we've learned a good amount. And so I'll say a little something about the conclusions and the take-home messages from these different papers. And then finally, many of you may want to know about what are the practical considerations I need to think about or take into consideration when I want to use chemically modified CRISPR in the lab. If you don't synthesize chemically modified oligos, where do you buy those, and what do you need to know, what to look out for? So I'll talk a little bit about that. Okay, so one of the holy grails of genome engineering was to be able to engineer the genome precisely, and this This was not easy to do. It could be done several years ago, but it was very expensive. Only a few labs had access to these reagents. So what I'm referring to is talons and zinc finger nucleases and meganucleases. These enzymes were around and developed, but they were really hard to implement.
I'm sorry,
Okay,
oh, there you
I think
are.
I'm back online. Our wonderful university Wi-Fi seems to have given out on me. So hopefully everybody can hear me again. And again, so the holy grail of precision genome engineering is to be able to do it at will, easily, extend this to engineering crops, industrial applications, obviously gene therapy in human patients. And when I was a postdoc, I remember we got a review for a manuscript, and that's shown here at the bottom. And the comment was that we should delete or mutate the seed sequence for our putative microRNA that was in the promoter region of our gene. And at the time, that seemed a little bit ludicrous because we knew it would take months and months of work and tens of thousands of dollars to accomplish this. So we had to kind of wave that off and convince the reviewer that, yeah, that's a good idea, but we can't really do it. But nowadays, if you get these sort of comments, you may not be able to so easily wave them off, and you may be obligated to do such experiments because of CRISPR. Let's see here. Okay. And so here is just an example of talon nucleases or talons. These are derived from tail DNA binding domains that you can link together, and you can make an array, and each of these little helix turn helix motifs can be designed based on the amino acid that you have in its loop, be designed to target a specific nucleotide. So if you want to design a talon that can bind a 10 nucleotide DNA sequence, you need 10 of these loops, 10 of these helix motifs linked together contiguously. Okay, and then of course you have to fuse this to a zinc finger or some other nuclease that can cut the DNA. And on top of that, it only cuts one strand, so you need two of these adjacent nearby each other. At least that's the last, maybe the technology is changing by now. So a bit complicated. And what CRISPR does is allow you to cut both strands with a single enzyme. And by the way, it's programmable with an RNA sequence. Okay, so this is a crystal structure of Cas9, and the protein is shown in gray. And here is the guide RNA that binds to the Cas9. And the guide RNA is a bit like a cofactor. It binds tightly. It's required for the activity of the enzyme. It guides the catalytic activity. And it also, in addition to binding the RNA, the next step is it will recognize double-stranded DNA, specifically a motif called the PAM sequence. And the PAM sequence is a small 2+ sort of nucleotide motif that is required to bind first. And then once it does, the guide sequence of the CRISPR RNA will pair up with the target DNA, and you'll get DNA cleavage if you have an active enzyme. Okay. So the beautiful thing about this is the guide RNA itself, just the sequence, is what dictates your substrate specificity. Okay. Again, this is also quite, if anybody's used this enzyme or tried to make it, we purified it in E. coli, and it didn't take long. It was very easy to work with. And the fact that it's programmable with an RNA, very similar to RNAi and Argonaut, it made it catch on very quickly. Okay, so you can do gene knockouts. That's the major thing we call gene editing. Create a double-strand break in your target gene. You'll get small insertions or deletions during the repair process if it uses the non-homologous end joining. And this basically constitutes functional gene knockout. You can also insert sequences specifically. This is a little lower efficiency, but allows you to do more precise genome editing. And just listed below, I mean, these are examples where this CRISPR is being used, a lot of synthetic biology, model organism development, and clearly for therapeutic applications, that's a major goal. Okay, so it is beautiful, it works great, but it has its caveats. One is efficiency. Efficiency, we'd like it to be 100%, but it's not. It's rarely does it reach that level. Excuse me. Also, it has some variability with depending on the sequence context. So, you know, one target might cut really well, another target not so much. Specificity, we have on-target cleavage shown here, which is what we want, but also you can have off-target sites that look very similar to the on-target site, maybe one or two mismatches, but you can get cleavage there. And so these obviously are bad. We don't want these off-target cleavage events. And also stability of the enzyme, how active, you want it to be active long enough to complete the on-target editing and do its job. But what happens when it sticks around for a long time or degrades too quickly? So would like to be able to control stability. The nice thing is that chemically modifying the guide RNA component of these enzymes can actually address some of these challenges. And so that's what we've seen in the literature. So what I'm going to do here is talk about why modify the guides. One is, like I just said, we can hopefully address some of the shortcomings at the biochemical or enzymatic level. The other is an area that I'm pretty familiar with from my background, and that is using chemically modified nucleotides to kind of probe structure, function, activity relationships and understand how these enzymes work, their dynamics, which are hard to capture just by crystallography. And then by learning more about the structure-activity relationships, we could engineer new novel RNA-guided systems, or it teaches us the basic principles of what we need and what works and what doesn't. And this is a more fundamental approach that goes along with or is complementary to the standard just screening oligos for the best ones that edit. Okay. And then finally, therapeutic development. You know, we know from many years of ASO research and siRNAs and trying to put those in the clinic that we really absolutely need to chemically modify RNA if it's going to be a therapeutic, at least by the delivery methods we would think like systemic injection or most other methods. CRISPR-Cas9 is a little different. You can always do a viral approach and express the Cas9 and the RNA. There, of course, you don't need chemical modification, but if you're going to be doing lipid nanoparticle delivery or formulations, it's going to be probably desirable to chemically modify the RNA and stabilize it. What's shown here to the right is this great idea, and I'll talk a little bit about the paper that this came from, which is Radar et al. In 2015. And this was an idea that was put forth that you could make a chemically modified CRISPR RNA that was a lot like an ASO, and so heavily modified and functional, and you could deliver that separately, and then you could combine that with a viral delivery of the gene itself, the Cas9, and maybe its other RNA component called the tracer RNA, and then you would have sort of an APO enzyme, a non-active enzyme poised to do editing, but still lacking that CRISPR RNA, which you could then add separately as a chemically modified oligo. And so I like that particular paper when it came out because it gave us at least one idea, one way that chemical modification would be really helpful for therapeutics. Okay, a little overview real quick to familiarize ourselves with the RNA guide. There are two components of the naturally occurring CRISPR RNAs, and this is for the Cas9, the standard sort of Cas9 CRISPR system. You have a tracer RNA, which is shown in blue, and that tracer RNA is like an anchor. It binds to the Cas9 stably, and it presents to a CRISPR RNA interaction region, or what's often sometimes called the repeat region. And this will base pair to another RNA called the CRISPR RNA, the CRRNA. And then the CRRNA, the other end of it, the 5' portion, shown with just purple ends, is a guide region that can vary. And this is how you program it, is you put in the guide sequence that you want based on the DNA target that you want to bind to and maybe edit, for example. Okay, so this is called the dual guide RNA system, the dual guide system, or the naturally occurring system. To simplify the system, the Doudna lab pretty early on, 2012, realized that you could fuse the CRISPR and trace RNA together, shorten them a little bit, put a GAA tetraloop on the end, and these artificial single guide RNAs, they work as well or better than the dual guide system. And I think really they were initially made to simplify genetic expression systems, because you would only have to express one RNA instead of two RNAs. Okay, the dual guide system was really out of use for the first few years, but once it was discovered that you could electroporate these in, or again, chemically modify them, or combine them with the protein, and you can electroporate the RNP into the cell, then they've gained they've gained more favor in recent years. Okay, the RNA that mostly gets chemically modified or has been modified in a lot of these publications I'm going to discuss has been the CRISPR RNA, which is 42 nucleotides. Several of these research reports have not bothered to touch the tracer RNA. Only a handful have really looked at the single guide RNA, because obviously it's longer and harder to synthesize. Okay, so I want to start out by talking about publications that I guess we'll start with simple and then move to more complex. And so the simplest modification schemes are really just to try and cap the ends of the RNA and protect them from exonucleases. In fact, the first sort of official chemical modification of CRISPR RNA paper was Hendel et al. This came from Matthew Porteus's lab, and he collaborated with Agilent, and this is in Nature Biotech 2015. And what they did is they just want to incorporate modifications on the termini, with obviously the goal of protecting from nucleases. So shown here in the end, at the bottom, is the 3 modifications they used: 2'-O-methyl, 2'-O-methyl with a phosphorothioate, and 2'-O-methyl with a 3'-thioPase modification. And so these are, I may use the shorthand throughout this talk as MS or MSP for these two, because they occur again. And then other modifications that were used for some CPF1 studies: 2'-fluoroRNA, unlocked nucleic acid, and locked nucleic acid, in addition to just normal 2' deoxy DNA. Okay. So the Hendel et al. Paper was great because all you had to do was add, it seemed, these few modifications on the end, the MS or the MSP, the 5' and 3' ends to cap. And this study was done with single guide RNAs. And what they found is it produced a significant improvement in editing efficiency. Now, in this particular publication, again, they focused only on the single guide RNA, and they were really interested in co-delivering the guide RNA with a messenger RNA for Cas9. So you can have like a DNA-free system and not have to worry about incorporation of the plasmid or extended expression of your Cas9 over time. Okay. So later, a similar publication followed, and this came from Dharmakon, and they were looking also at single guide RNA, and they were investigating the extent to which you have to chemically modify. And they found that they only needed one 2'-O-methyl phosphorothioate at the 5' and 3' ends of the single guide RNA. And this gave a moderate improvement in efficiency. For the dual guide system, they found that two of these MS modifications were ideal, and they gave sort of the best activity, lowest toxicity in cells. And this was particularly when you were trying to co-deliver with the mRNA. Now, they pointed out and spent a lot of time in this manuscript pointing out that there was really no significant benefit if you were assembling the RNP first and then electroporating it. And that makes sense because the Cas9 does a really good job of protecting the RNA so it doesn't have to get degraded. Okay. Move to the next slide. There have been some CPF1 studies. 3 CPF1 papers have come out, and these 2 of them. In the first paper, they did try to do more extensive chemical modification, and also we'll revisit this paper a little bit in a future slide here. But what they found was that you could successfully add 2' fluoro up to 5 2' fluoro RNA nucleotides at the 3' end of the guide, and this is not in the seed region. And so you can see here, this is the sort of schematic for CPF1. Its structure is a little different. The CRISPR RNA is just a single RNA naturally. It has a 5' pseudoknot structure, which is referred to often as the 5' handle that binds the protein. And then the guide sequence is actually at the 3' end, and the seed region is right here at the 5' portion of the guide sequence. Okay, so what they were doing is they're putting modifications here. They found that it could tolerate 5 2' fluoro pretty well. They tried to put modifications in the 5' handle, but nothing was really successful. So the conclusion is that the 5' handle is not easily modified. Another paper, this is a pretty recent paper. This publication really wasn't focused, this is the Park et al. 2018, wasn't really focused on chemical modification. And instead, they were just putting a little 5' extension on that handle, and they found that this really improved editing efficiency inside of cells and improved transfection efficiency. But in that paper, we need to mention it because they added MS, 2' deoxy, and phosphorothioate modifications in that extra extension, and this helped. This didn't reduce the activity, so it showed that it was tolerated. And this really significantly helped with serum stability or nuclease stability. Okay, so DNA may or may not be considered a chemical modification, but in a few papers it was actually used as a modification, primarily to see if it could improve activity, reduce off-target effects, or as a probe to see what could be tolerated. We know DNA is very affordable, has pretty predictable properties, so I think it was a good choice for some of these manuscripts. Okay, so the guide RNA makes 2' OH specific contacts with Cas9. The question was, how important are these contacts? So DNA is ideal for this sort of study. So substituting RNA nucleotides with DNA can probe the requirements for these contacts, and also it was found that you can modulate activity and specificity with DNA. You may notice too that I'm trying to present these papers in sort of the order in which they were published, not necessarily in the order of what I think is most important or not. So Ruida et al. Was the first one to come out really that investigated DNA. And what they did is they replaced all the nucleotides in the CRISPR RNA with DNA, and then there's 6 central and 1 7th terminal nucleotide that according to the crystal structure make 2' hydroxyl contacts with the Cas9. So they, in the context of an otherwise all DNA CRISPR RNA, those ones are left as RNA and then systematically swapped in DNA bases to see kind of which one was the most important. This study was primarily in vitro. In fact, when they tried to put these CRISPR RNAs in cells, they did not work for editing. That's something we and others have observed, that too much DNA is not good, and then these positions are really critical. So certain RNA residues with known 2' OH contacts could not be replaced in this all DNA guide. They also did a nice little binding study, single molecule binding study, that showed that incorporating the DNA into these CRISPR RNAs reduced the binding to the target, so sort of modulated target binding, which turned out to be beneficial in the next study. In this next study by Yin et al., this was in early 2008, the idea was looking for ways to improve or reduce off-target editing and improve the specificity of the enzyme. The rationale was that DNA doesn't quite bind as tightly, DNA versus RNA DNA. So putting some DNA bases into the CRISPR RNA sequence particularly in the guide and particularly not in the seed region, could potentially give you lower off-target editing. And so this is just an image from the paper giving an example of how they moved along and systematically added in DNA bases. So the first approximately 6 to 8 nucleotides could be easily replaced with DNA and not really compromise editing efficiency. But of course, as you moved into the seed region, you lost all your activity. Okay. What they did find, they went on to show that modification, DNA substitution could also be done in the 3' end of the CRISPR RNA, and that was tolerated to some degree. And then you had reduced off-target editing, a little bit better specificity with DNA substituted CRISPR RNAs. And then the last one in this series I'll talk about is one that came from my lab. We published this 2018. This was an entirely in vitro study. We do have quite an amount of cell-based editing that's unpublished still. We're trying to figure out basically why too much DNA or DNA in certain positions compromises editing efficiency so dramatically. But in this particular study, we saw that DNA, particularly in the trace RNA pairing region, could dramatically and significantly improve the editing efficiency and the kinetics of the enzyme. And again, this was in vitro. We found also too that you could swap in a lot of DNA bases, or nucleotides, excuse me, into the tracer RNA, and those were tolerated. And we also did some binding studies and found that the DNA does inhibit target binding, but also can improve specificity of the enzyme. Okay. So now, a couple papers were looking more at where can you put modifications internally. So moving beyond just capping the ends or simplified substitutions like DNA, can internal modifications be tolerated? Where can you put them? And can they be used to sort of tune the activity of the enzyme? So the first one published in this sort of respect was Ryan et al. And this it came from the Agilent team who had collaborated with Matthew Porteous for the Hendel et al. Paper. So this is sort of a follow-up paper where they're not just trying to cap the ends, but they want to systematically place MSP modifications through the guide sequence to see what was tolerated and what was the effect on the activity. So surprisingly, they found a dramatically enhanced specificity when they put the MSP at specific positions, in particular position 5 and 11. They proposed that this modulates the conformational transitions during catalysis. And this makes sense because position 11, near the middle of the sequence, is right flanking or right there near the seed sequence. Cromwell et al. Looked at DNA NC, so bridge nucleic acids, and also locked nucleic acids, and they found that if you put these, and they often had 2 or 3 contiguous next to each other, in the central portion of the guide region, you can actually improve specificity and reduce your off-target editing. They also did some single molecule studies and proposed that the enzyme kinetics were slowing down, and this was reducing target binding, which was providing that little window of specificity. Okay, so this is a common theme that's emerging. If you want to improve specificity, you need to sort of dial back the binding affinity to the target, or you need to somehow modulate conformational transitions of the enzyme. Okay. So that was with Cas9. There was some modification done with CPF1. I mentioned the Lee et al. Paper previously. Now they did try to do more extensive modifications, especially in the 5' handle, and these were absolutely not tolerated with the particular designs they did. They were trying to replace entire sort of chunks of the 5' handle, and it just was not tolerated. We also found the same thing in the McMahon et al. Paper, which was in early 2018. Modifications in this 5' handle are not well tolerated. This can be easily explained because the 5' handle forms a very unique pseudoknot structure. It's forms a stem, and then it folds back and interacts with the loop of the stem. And then the CPF1 sort of wraps around this really nicely. So it goes without saying this is a very very, very sensitive structure. However, there were some interesting points with McMahon et al. And also point out that McMahon et al. Is sort of a follow-up too from an earlier paper from this group. This is the Ionis Pharmaceuticals group in collaboration with Don Cleveland and Radar. And so they had an early 2015 paper, which I'll get to. But they're trying to do more extensive modification of CPF1. The problem is CPF1 is just not easily modified. These CRISPR RNAs do not tolerate modification the way the Cas9 CRISPR RNAs do. Something I thought was interesting is they had made an entirely all PS backbone CRISPR RNA that still showed 30% activity. So this is encouraging because even though you can't remove all the RNA bases, you may be able to remove a number or substitute a number of them, and then for those that remain, you could maybe use PS linkages. And the take home really that I took from this paper was that 2' fluoroRNA and some other modifications, 2' methyl a little bit, were tolerated a little bit at the far 3' end of the guide region, outside of the seed. Okay, so this kind of confirms what Lee et al. Had seen also previously. Okay, now I want to move to what I call very extensive or complete chemical modification, or attempts to completely remove all the RNA bases or really explore the chemical space a little bit. And I guess a motivating question for these studies would be, what are the limitations to complete modification? And so some of these studies used the structure of the Cas9 to try and guide their modification schemes. Some did sort of traditional, just let's modify blocks or chunks or do certain patterns to see what effects we get. The first one published was Radar et al. And as I just mentioned, the McMahon was sort of a follow-up of that, but on CPF1. And this was a great paper. I felt like, at least for the field, this really early field of CRISPR chemical modification, this was kind of seminal because they did a lot of modification. They used fairly standard modifications. The CET is not standard, but this is an Ionis Pharmaceuticals custom, very similar to LNA. And they did it in an all-PS phosphorothioate backbone. And they found that they could get good editing as well or better than the single guide RNA or the all RNA version that they were using. And also they were able to shorten the CRISPR RNA. So they were then proposed, I think the novelty here was they proposed a new way to combine synthetic CRISPR RNAs with viral approaches. And this is what I really liked about the paper. Nobody knows if this will work or not, but it gave us a clear idea of how you could use chemically modified CRISPR RNAs maybe in the future, one of many potential ways. And that is you could first express the Cas9 and the tracer RNA from a virus, and then follow with a chemically modified CRISPR RNA delivered much like you would an ASO. And then only as long as the ASO is being given or administered will you have editing, and when you withdraw the CRISPR RNA, you will not get editing. Okay, so like that study, one of the problems was they could not put modifications in the seed region, only outside of the seed region. So one of their conclusions was you just simply cannot modify the seed region. It was at the time that was a good conclusion, but it was limited based on what they tested. And we now know that you can modify a lot of the guide region, as some of these studies show us, but you have to avoid certain positions. Okay. The Yin et al. Paper was the next one published. This came out in 2017. It's Nature Biotech. This was a single guide RNA attempt to chemically modify the single guide RNA completely. They ran into some issues. I mean, obviously some positions couldn't be modified, but what I'm showing here to the side is sort of their final design, what they call an enhanced single guide RNA, with extensive 2' methyl modifications, a lot of phosphorothioate in this 3' protein interacting portion, and then 2' fluoro and RNA primarily in the seed region. They used a crystal structure to try and guide the design. So it's a couple positions, position 2, and then 5 and 6 here. in the guide region, those are known to have 2' hydroxyl contacts with the protein. But those, when they tried to modify them, they lost activity. And then they went on with a nice thing about this study, they went on and used this to do lipid nanoparticle delivery of the single guide RNA and do editing in mouse liver. So
Are you back on?
yeah, I'm back on. I apologize,
Okay.
guys. Something's funny with the Wi-Fi, but we should be able to finish the talk hopefully without it dropping again. So I was just saying about this unit all paper that I liked that they went into mice and they showed editing and that they got improved editing with this chemically modified version. Another paper that I really liked was Miradol. This came out of UMass. It's a collaboration with John Watt's lab, Anastasia Korbova, and Eric Sontheimer. And they use sort of off-the-shelf modifications, the 2' fluoro, 2' O-methyl PS that we're seeing a lot of. But they did a really good job of systematically narrowing down what positions could and could not be modified. And they removed all but a handful of RNA nucleotides in the CRISPR RNA, and those correlated with the CRISPR RNA nucleotides that are known to make 2' hydroxyl contacts. So you should see a theme here with positions you can't modify. However, they added PS linkages to those RNAs. So technically, they've achieved complete modification of the CRISPR and trace RNA. Combining these two completely modified versions, though, you saw a significant decrease in overall activity of the enzyme efficiency. But nonetheless, it was this and the Unidol paper were sort of a tour de force example of how you can go through and systematically reduce the RNA bases. The O'Reilly et al. Paper came out the end of last year. This was a collaboration with Masad Dama and myself. And our goal was to test a broader variety of more exotic modifications to see what would be tolerated, what would not be tolerated. It was more of a structure, function, activity relationship study. And we're looking for general rules about what can and cannot be tolerated. One of the take-homes was that you needed to maintain A-form-like structure in the CRISPR RNA, both in the guide region where it's going to be pairing to the DNA target, but also in the tracer pairing region where it's going to be base pairing with the tracer RNA. This was a dual guide RNA study, and mostly in vitro. We did some editing at the end with a select few oligos. We also found that bulky moieties, bulky sort of modifications weren't really well tolerated, maybe in certain positions. But the take home was A-form helical structure was important. And in fact, in vitro, I think it's now commonly accepted, in vitro, you don't need any RNA bases in the CRISPR RNA to get fantastic cleavage activity. You can completely convert the CRISPR RNA to all 2' fluoro, for example, and it gives great activity in vitro. But when you put it inside of cells, the editing is inefficient. So this is an area that still needs some investigation to figure out what's the discrepancy between in vitro and inside of cells. And finally, IDT, they sell some Alter CRISPR reagents. These are known to have extensive 2' methyl modifications, some terminal PS linkages. Not published it, but they've disclosed this and talked about it at talks and at meetings. And I'm sure maybe Mark Belke or some others might be happy to tell you more about them. But you can buy these directly from IDT. Okay, so that really covers all of the chemical modification CRISPR papers that I'm aware of. There may be some other minor ones. There are some minor ones, but so there's not a lot of literature, and that's why I thought it would be good to just kind of go through them all in this webinar. We have some take-home messages, take-home summary that I'll just go through briefly. You can increase your editing efficiency and your nuclease resistance with modifications. Some of them can even enhance biochemical activity, especially DNA, surprisingly in certain positions. Some of these modification schemes, depending on where you put the modifications or what modifications they are, can enhance specificity, reduce off-target effects. We also know that chemical modifications, because you can increase the binding affinity between a CRISPR and tracer or certain stem structures of the guide RNA, you can actually truncate or shorten the CRISPR RNA. And a paper that I didn't talk about that came out 2018, this was an IDT paper, Schubert et al., 2018. They studied the effect of modifications on interferon responses. Chemical modifications are going to be key for doing non-viral therapeutic approaches. And then I think one of the themes that came out is that although you can modify most of the nucleotides in a Cas9 guide RNA, some of them are really recalcitrant to modification. So you could almost make an RNA-free CRISPR, not quite. And then unfortunately, CPF1 guide RNAs are even less tolerant of modification, and they're going to require a lot more work to probably modify and stabilize. And then one of the take-home messages from work that I was doing with Mossad and our DNA study was that A-form helical structure overall should be maintained. So modifications should be incorporated that maintain A-form structure ideally. Okay, so if you're wanting to use chemically modified CRISPR RNAs or single guide RNAs, where you get them, I'm just going to highlight 3 companies that sell chemically modified CRISPR RNAs. There are others, and there may be more to come. They're all proprietary. They're not going to tell you exactly what they use or where they put them, but some of this information can be known from their publications. So the Alter CRISPR system I've used before. It works great, no complaints. We just prefer to make our own versions and synthesize our own with Mossad. But it's primarily 2'-O-methyl with some phosphorothioate. There may be some 2'-fluoro in there. But they can also custom synthesize anything you want. So I often will order CRISPR RNAs from IDT with 2'-O-methyl or fluoro or phosphorothioate because it's expedient and quick to do so, and they'll make them for you. Okay. Also, the CRISPR RNAs, IDT will make up to 60 mer for the RNA, and all you have to do is desalt. So I think it's quite affordable, and they give good quotes, so I would check them out. Synthego is a company that their sort of flagship product is synthetic single guide RNAs, so chemically synthesized. These are not chemically modified, although they will chemically modify them for you, and they like to brag about their chemically modified single guide RNAs. So I've not tried these. My impression is they do work very well. They try to make it affordable, so you can get free samples. They don't send you much, 1.5 nanomoles for $225, but that's actually enough to to do several transfections and several treatments and decide if you like it or it works or not. So a good company to go with. They can also do custom synthesis of the single guide RNAs, but I think you'll need to contact them and find out what they can and cannot do for you. So not bad. They do charge also less for academics than for industry. Okay, Dharmacon. I mean, I was ordering RNA from Dharmacon when I was a grad student, and they've been in the synthesis business for a long time. And so they also now they sell synthetic CRISPR RNA, tracer RNA, single guide RNA, and that they do have proprietary chemical modifications that I don't tell you exactly, but you can deduce from their website and from the publications that they're just putting 2'-O-methyl phosphorothioate caps on the ends of the RNAs. And that's their chemically modified version. I've not looked into the price, what they charge. Dharmacon also does do custom synthesis, so you can check them out. Okay, so I would just like to end off a little bit speculation here, if you will, about chemical modifications of CRISPR and their use in therapeutics. It's clear that they will be useful for things like ex vivo editing, or if you want to deliver the guide RNA with the mRNA of CRISPR by lipid nanoparticles, for example. Those are going to be great opportunities to chemically modify. How much chemical modification, or where you want to put them, or what you're trying to achieve can vary, and so that will determine what kind of modifications to do. Also, too, if you're doing viral delivery of Cas9, you're never really going to need to do any chemical modification unless you want to do this viral synthetic combination that was proposed by Radar et al. In 2015. Depending also on what the goal is for the therapeutics, there may come a need to sort of resolve this issue with remaining RNA bases. So that's a good question. Are there mods out there that can compensate for RNA at these key residue positions? So that may be worth investigation. Also CPF1, if we want to use CPF1 for therapeutics and do chemical modification, so far there's been investigation of at the 5' and 3' ends, but that 5' handle in particular is really tough to try and modify. So probably there'll be some more work that will need to be done there for CPF1 CRISPR RNAs to be used. There are other therapeutic or drug-related applications for CRISPR, like non-editing approaches. And what are some other things that you could use chemically modified CRISPR RNA or CRISPR RNA sort of mimics for? One direction that my lab's been investigating is actually using mimics sort of the CRISPR RNA as competitive inhibitors for CRISPR. So there's creative ways to use chemical modifications, and I think that they're going to gain more traction. But the handful of publications we've had so far have told us a lot about what does and does not work. So now it's yet to see to what extent they'll be used for therapeutics. Okay, so with that, I'll let you guys ask some questions. I think I see a couple there already. Okay, Din said, hi, will it be possible to access the recording of this webinar later? And the OTS webinar says yes. So yeah, it'll be uploaded and available according to Petra. And there's other ones that are available. What's that?
Thank you very much, Keith. That was a very good talk. So if anyone has any questions, please type them in the chat box, and Keith will do his best to answer them.
Mm-hmm.
Oh, I think you must have answered all the questions in your talk. Oh no, there we go.
Because I talk a lot. There are other webinars that are uploaded too. If people have bothered to look online, they can see. So someone says, Vivek says, can I comment on correlation between melting temperature and CRISPR-Cas9 activity? That's a good question. So if you're talking about the melting temperature or the TM of the CRISPR RNA itself, I'm assuming you may be talking about the guide region and it's pairing to a target. That's something that I don't know has been fully investigated, but it's investigated in other literature outside of chemical modification. But there may be some correlations with activity. If you're binding too tightly to the target, a lot of GC content, for example, that may be a problem with off-target. And so that would be, maybe adding some DNA at the termini or some modifications that reduce your affinity slightly or your TM. That may actually help reduce off-target editing. And I believe there's a window which you're going to want to try and operate in to maintain on-target editing. So let me know if that doesn't answer your question all the way. What would be the best assay for CRISPR specificity? Well, you can do, if you're just doing in vitro, you can look and see what we did in the Carchi et al. Paper, where you basically just take a synthetic target and you will incorporate 1 or 2 mismatches. I like this approach because you can really control where mismatches are and what your target sequence is. So that's in vitro, and then you just measure, cleavage activity. For cell-based editing inside of cells, you can do small scale. If you have specific off-target sites you're looking for, you can then PCR amplify those up and do tied analysis, which is a Sanger sequencing-based method, and look at, what's the % editing I'm getting at these potential off-target sites. So, so that's what I'd probably recommend for that. If you have a broader list of off-target sites, you can, something actually we're thinking about trying is IDT has a ramp-seq method where you can, it's designed to be coupled with next-gen sequencing, but you can incorporate dozens or hundreds of potential off-targets, PCR amplify those, and next-gen sequence those. And then finally, if you don't know what you're looking for, or you just want to look for all potential global off-targets, then you're going to want to do a next-gen sequencing, whole genome sequencing. I have not done that, and I don't want to comment on which one's the best one. So you'll have to just see what works and doesn't for you. So how many LNAs in continuation can be incorporated in CRISPR-Cas9? A good question. I would refer you to the Cromwell et al. Paper. Let me check and see if it was the Cromwell paper where they did the BNA and the LNA work. Let's see here. Yeah, so Cromwell did BNA and LNA, and they were putting 2 or 3 next to each other in a row. And that's where they were seeing the best sort of specificity improvement. In particular, when these were placed sort of in the central portion of the guide region and as part of or flanking the seed region. So how many can you put in there? I don't know. Obviously, LNA and bridge nucleic acids have a huge impact on your melting temperature. So you might actually cause higher off-target effects if you put these in the wrong region, or you lose activity if you put them in the wrong region. I mean, maybe at the near the 5' end of the CRISPR RNA. You might kill activity if you put them or too many of them in the seed region itself because of those critical 2' hydroxyl contacts. So how many can you put in? I don't know, a couple. But I would refer you to that paper to see what they did more specifically. So someone says, I'm going to use saCas9 and it requires a different tracer RNA compared to SbCas9. Are there any companies that can provide guide RNA for saCas9? Yes, there are. You can just design your own and order it from IDT. Synthago, I believe, makes tracer RNAs, CRISPR RNAs for different enzymes. Not a lot's been done testing other Cas9s. However, in the Karchi et al. Paper, which was the DNA substitution paper from my lab, we did test these effects on saCas9 and saw the same trends. And what we were looking for is that enhanced activity when you placed DNA in there. So yeah, saCas9 should work well. What modification patterns to use, I don't know, but if you just want to stabilize saCas9 CRISPR RNA, I probably would just start with a couple, maybe 2 terminal PS mods, PS 2' methyl modifications, and I would refer you to the what's it? Let's go back a little bit here. Probably the Bacilla et al. Paper and the Hendel et al. Paper. I would refer you to those probably. So I do. Oh.
Right, I think one more question coming. No?
Maybe. Well, as I say,
No.
I do know that, I mean, just from hearsay, there are a number of companies, the CRISPR companies that are out there who are doing lipid nanoparticle or formulation delivery. They are playing with chemical modifications, and they're very much aware of it. So this is in the therapeutic space and for therapeutic applications. But for us academics and in the community at wide, we're not always aware of this because unless it's published. In fact, same with DNA substitutions. We know they're helpful and they work, and there's more work being done out there, but some of it's not published. In fact, some of our work still we're trying to finish up and publish. So hopefully more of that will come out and we'll learn more as time goes on.
Well, there's no more questions, then thank you again, Keith. That was a really great talk. Just for everyone, keep an eye on the website. There should be more details soon on our next webinars, and they should be on the design of microRNA therapeutics and another one for aptamers for targeting. Thank you very much,
Thanks,
everyone.
Yep.
Thank you very much, Keith.
Yep.
See you. Yeah. Bye.